This is part 1 of 2 on Chapter 10, Genetic Engineering. In this part of the chapter, we're going to look at different tools of genetic engineering, and then the second part, we're going to actually use those tools to see what we can do with the different tools and techniques that we have. So genetic engineering, it's a type of applied science, and applied science is just using basic science knowledge that we have discovered and that we know a lot about to make useful products that we can use for different applications. And usually these useful products are for something that's going to help humans in some way. So there's bioengineering, which is where we do a direct, deliberate modification of a genome. And we also have biotechnology, where it uses an organism's biochemical and metabolic pathways for industrial production. So bioengineering is we can modify genomes, take one gene from one organism and put it into another organism, for example. Biotechnology, the example I'm going to go through is making insulin. So we need insulin to treat diabetes, so we can actually use a real organism to produce insulin for us, and then we can bottle that and put it through the pharmacies to help people. Okay. So we're going to use different things that we've talked about and see how we can actually apply these concepts to help humans do something that's useful. So this section, like I mentioned at the beginning, so we're going to look at what tools and techniques are used in genetic engineering. So these are just tools and techniques. So you have to understand these first, and then we're going to use them and show you different ways we can use them in the next part of this chapter. So one tool that we can use is called a restriction endonuclease. Okay, so restriction endonucleases, these are enzymes that recognize specific sequences of DNA. So they're enzymes that can identify parts of DNA and then break the bonds between these adjacent nucleotides. So these enzymes are used for dicing DNA or splicing DNA, and then we can hook different DNA from two different organisms together using these restriction endonucleases. So these enzymes are used in lab to cut DNA into restriction fragments. And there's some examples right here. So the endonuclease, we have 3 going across the top. So EcoRI, HIND3, and HAE3. So these are different enzymes. They recognize certain sequences in DNA. So the EcoRI, it looks for GAATTC. And then this enzyme is going to cut between the G and the A on the top, and then the G and the A on the bottom. So the dotted line represents the cutting pattern that it's going to cut in the DNA. Same with the other 2 endonuclease enzymes. They have different cutting patterns that you can see here. So the restriction endonucleases are the enzymes that do the cutting, and the cutting creates restriction fragments. So these restriction fragments, some of them have what are called sticky ends, where they have a little overhang on one side of the DNA compared to the other side of the DNA. So if you look at the pink and the blue DNA in the middle of this image or slide, on the top right you have a little pink. The pink is longer than the blue, and then on the bottom the blue is longer than the pink. So you have these little overhangs, or they're called sticky ends. So what we can do is we can use another enzyme called ligase. This ligase enzyme is going to rejoin the bonds on those sticky ends that were created by the endonuclease enzymes we just looked at. So ligase allows us to insert DNA from one organism into a DNA chromosome of a second organism. And you can see that on the bottom of the slide. So we have a yellow piece of DNA that we cut out from organism 1, and we're going to insert it into organism 2, which is the green piece of DNA. And then the ligase is going to hook the 2 ends together so we get one continuous piece of DNA. Okay, other types of enzymes we can use, there's an enzyme called the reverse transcriptase. This makes a DNA copy from RNA. So if you remember from chapter 9, we talked about transcription. Transcription is where you take DNA and you make an mRNA from it. Reverse transcriptase, you're just going in the opposite direction. You take your RNA molecule and you make a DNA copy out of it. And it's called a cDNA molecule. So cDNA can be made from any of the different types of RNA. So we talked about mRNA, the messenger RNA, tRNA, transfer RNA, or the rRNA, the ribosomal RNA. So you can take any type of RNA and go backwards to make a DNA copy of it. So why this is useful, it provides a means for synthesizing eukaryotic genomes from your mRNA. And in eukaryotic cells, remember, your pre-mRNA goes through processing. So if we go from the mature mRNA, we make a DNA copy from it. Our DNA copy only has the exons in it. It's free of introns. So we're kind of taking out all the introns, all the extra DNA that we don't need to begin with anyways. And this is showing this process. So normally at the top we have a eukaryotic piece of DNA at the very top, and it has exons, the red parts, and introns, the blue parts or purple parts. So remember, during transcription you take your DNA and you make a pre-mRNA molecule. So that's the yellow and purple alternating molecule. That pre-mRNA molecule has to go through processing, so it has to be spliced. You're going to remove the introns from it. And you end up with an mRNA, a mature mRNA that only has the exons. So that's in the middle, the yellow line. So we've cut out our light purple introns, we only have exons. So what reverse transcriptase can do is we can take our mature mRNA, that yellow goldenrod molecule, we can put it in a tube with the enzyme reverse transcriptase, and that will create a DNA molecule from it. And this DNA molecule, called cDNA, is free of all those introns, those extra pieces that we don't need. So we get a piece of DNA that only has the genes that are going to be expressed. So we cut out, we don't have any of that junk left over. So those are a few enzymes that we can use, different tools and techniques. Other techniques that are used in this genetic engineering is called gel electrophoresis. Gel electrophoresis, it can separate out DNA fragments based on their size. So you have different DNA samples, you cut them up with different restriction endonucleases, so you get cut up pieces of DNA. You take that sample, you place it into a soft agar gel, and then you're going to run an electrical current down the agar. So how this works, the DNA naturally has a negative charge to it. So we're going to hook up a negative charge at the top of our agar box, and then we hook up a positive charge at the bottom of the agar box. So our DNA has a negative charge, it's going to move towards the positive charge, so it's going to move towards the bottom of the box. And the cool thing about this is that smaller pieces of DNA are going to move faster through the agar because they can get through the agar. The larger pieces of DNA are going to move slower through the agar. So we're going to separate out the different DNA pieces based on their size. So small ones at the bottom, larger ones are going to stay closer to the top. Alright, so I kind of went over this already on the previous slide, but the negative charge of the molecule causes DNA to move towards the positive pole. So remember, DNA is negatively charged. It's going to move towards the bottom that has the positive charge because your opposites are going to attract. And this rate of movement depends on the size of your DNA fragment. So larger fragments move more slowly. They're going to stay at the top of the agar. Smaller fragments are going to move faster. They're going to travel further down the agar. Then once you run this gel for, it's generally about 40 minutes, all of your pieces of DNA are going to separate out. And we can't see these DNA samples with just using our eyes, so we have to stay in the fragment so we can actually observe them. So we have to use, and this method is useful in characterizing DNA fragments and comparing for genetic similarities. So we'll get into this more later. But you can see that going across the top of this gel, you have 1, 2, 3, 4, 5 different samples. The fifth sample is called a ladder, so it has known DNA fragment sizes in it that separate out. And then your samples, you can see you end up with different banding patterns in your agar gel. So this is how we just separate out these DNA pieces from each other. Alright, another method of analysis of DNA, so single-stranded DNA can unite with other single-stranded DNA or RNA. And then RNA can unite with other RNA. This is called hybridization. And this is the foundation for something called gene probes. And gene probes are used for detecting specific nucleotide sequence in an unknown sample. So if you want to see if you have this unknown sample of DNA, and you want to see if you have a specific nucleotide sequence such as like ATTA. You have this genetic probe, it's going to find the ATTA, hook on there, it's going to hybridize with your unknown piece of DNA. One really cool property of DNA is that when you take a piece of DNA, so you have a piece of DNA on the left side of the image, when you heat that DNA up to 90 to 95 degrees Celsius, so not quite boiling, but almost. When you heat up DNA, it causes all of the hydrogen bonds to break in your two complementary strands. So heating up a piece of DNA causes your two strands to separate. So we separate our blue strand from our pink strand in this example. So heating causes, again, those hydrogen bonds to break. And then when you cool that DNA back down, those hydrogen bonds reform, and the two strands are going to come together. And we're going to use this process in a certain technique, and I'll show you that in a little bit. So we can use this technique to sequence DNA. So DNA sequencing is just where we take a piece of DNA and we determine the actual order and types of bases in that piece of DNA. And the most common sequencing technique is called the Sanger technique. And I'm not going to go through it, but you can do this DNA sequencing so you can actually figure out if it's A, T, T, A, G, C. So you can actually figure out those base order using DNA sequencing. Another method with DNA is called the polymerase chain reaction, or PCR. And this is a method used to amplify DNA, so you can take a little bit of DNA and rapidly increase the amount of DNA in a sample. And it only takes a couple of hours. You can go from a tiny, tiny little amount to a larger amount that you can actually work with and possibly sequence or use to identify individuals. So PCR is a very important technique. We see it on like CSI all the time. They always do PCR to identify the victim and to identify the different suspects. So essentially, PCR is important for gene mapping, so looking where genes are on chromosomes. It's important for the study of genetic deficits and cancer, so looking at genes, again, identifying those cancer genes. We can use it in forensics, so identifying the victim or suspects at a crime scene. Taxonomy, looking at how organisms are related to each other, and evolutionary studies. So again, looking at how organisms are related to each other, how closely they're related to each other. And the polymerase chain reaction, it uses that heating and cooling method. So you go through multiple cycles in a polymerase chain reaction. So you start off with a segment of DNA that you want to amplify up at the very top. So we have our pink and blue DNA strand. You heat up that strand, which causes the two strands to separate from each other. So the pink and the blue are going to separate from each other. You put on primers, and then you make the complementary strand. So basically you take one piece of DNA, you force it to separate, and you force it to go through DNA replication that we talked about in Chapter 9. So you start with one piece of DNA, you'll end up with two, then those two pieces of DNA are going to go through another cycle and you'll get 4, and then 8, 16, 32, 64, and so on. So basically you're doubling the amount of DNA every time you go through one of these PCR cycles of heating and cooling.